Good evening. We'll be waiting for a couple of minutes so that everybody can join in. We'll wait till 6:05 and then we'll start.
Hello. Hello. So good evening, sir. So can you hear me, sir?
Yeah, you're audible.
Uh,
Uh, okay. Did you ask anything? Actually, there's a network issue here. Yeah. Is there any question?
am I audible, sir?
Yeah, you are.
No, I'm not unable to.
Can you see the
Uh,
presentation? Wait, it's loading. I'm just having some network issue. I'll try to solve it.
so
Uh,
actually, can you hear me, sir?
yes, I can. Can you hear me?
Yeah, sir. Actually, I'm student of from St. Pius Degree College, sir, from Hyderabad.
Yes, yes.
So actually, I couldn't submit my assignments due to my placements, sir. Today, I've joined only today.
Okay.
So could I write the exam? Like,
I think there are already the rules that they are mentioned regarding the assignments. You can, I think, miss a few assignments. Apart from that, you can use the section that is given to you in the course. I think it's the ask a question section. There you can ask this, and I think they will reply to you regarding what needs to be done.
okay, sir. I'll just. Okay, sir. Thank you.
I'm just trying to share this. Just wait for a moment.
Yeah, sir.
Yeah, is it visible?
Yes, sir.
Okay. I think we should begin since we don't have a lot of people here today. Okay, so welcome to the eighth session of the problem solving assignment of the course Genome Editing and Engineering in NPTEL. This course is taken by Professor Utpal Bora of IIT Guwahati, and I am Sanket Mukherjee, a PMRF fellow from the School of Biological Sciences in IIT Delhi. So we'll be solving few questions today. Let's see. So thalassemia is a hereditary disorder and it is inherited by A, autosomal dominant manner, B, autosomal recessive manner, C, decreased synthesis of alpha chains, and D, decreased synthesis of beta chains. I'll let you all have a minute to think about it and decide for the answers. Then we will discuss. Okay, so the answer would be thalassemia is a hereditary disorder and it is inherited by autosomal recessive manner. So let's talk a little bit about thalassemia. It's an inherited blood disorder which is caused when the body does not make enough of a protein called hemoglobin, which is an important part of red blood cells. When there isn't enough hemoglobin, the body's red blood cells, they do not function properly and they last shorter periods of time. So there are fewer healthy red blood cells traveling in the bloodstream. Now, red blood cells carry oxygen to all the cells of the body, as we all know. And hemoglobin, which carries this oxygen to all cells in the body, is made up of two parts called alpha and beta. So these are the two chains present in hemoglobin. And when there is thalassemia, it could be either alpha or beta. So this refers to the part of hemoglobin that isn't made. If either the alpha or beta part is not made, there aren't enough building blocks to make normal amounts of hemoglobin. So low alpha is called alpha thalassemia, whereas low beta is called beta thalassemia. So if you see about thalassemia, it's a hereditary disorder. It is inherited from a faulty, gene from your parents to the offspring. That's the idea. It's autosomal recessive disorder. So that's the answer to the question. It arises due to defects in hemoglobin production. It is different from other hemoglobinopathies like sickle cell anemia, which is caused by structural defects. Thalassemia is caused by decreased synthesis of alpha or beta chains of hemoglobin. So as you can see in the right, a person can have the wild type alleles where there is no mutation, whereas if there is a mutation for thalassemia, that person is called as a carrier. A carrier is not affected by the disease, but a carrier can potentially transfer the disease to the offspring. So that is what is given. Now, regarding the symptoms, it's very clear. It's anemia. So that happens due to loss of red blood cells, as simple as that. So there is shortness of breath, cold hands and feet, pale skin, irritability, dark urine, pounding, heart palpitations, and fatigue. There is iron overload that does not happen due to thalassemia alone, but thalassemia patients, they have to undergo regular blood transfusions. So what happens when there are regular blood transfusions? There is accumulation of iron in the body. So that causes problem. Uh, so there could be infection. Since they are getting, transfused bloods, there are, immune problems that arise due to this. There could be bone deformities and enlarged spleen. So these are the symptoms of thalassemia. Now talking about the question number 2. So to combat these issues, especially the iron overload, gene therapy is used, to prevent that. So question number 2 is, how many functional genes are there in beta globin gene cluster? Is it A5, B4, C10, or D6? I'll be giving you a minute to think about it and then we'll discuss. Okay, so the answer to this question would be A5. So there are 5 functional genes. Let's look at it. So let's talk about hemoglobin. So it's a heterotetrameric oxygen transport protein. So there are like tetramers uh, which are present. So again, these tetramers are formed of 2 alpha chains and 2 beta chains. So they are not homotetramers, rather heterotetramers. So they contain 4 polypeptide subunits of molecular weight 64.5 kilodaltons. 2 alpha and 2 beta chains of 141 and 146 amino acids respectively are found in the adult hemoglobin. The secondary structure of the polypeptide chains in HbA are alpha helix without any disulfide bonds and isoleucine. Also, beta strands are not present in HbA. So it's totally all about alpha. The alpha chains are present. The alpha and beta chain of HbA are almost similar. However, beta chain contains 8 helical fragments designated as A to H, while the alpha chain lacks helix D. Now, each polypeptide chain of HbA, it forms a 3-dimensional conformation, which is called as the globin fold. Now, if you see the structure closely on the right, so they have a heme group that contains the iron, right? And that is important to carry oxygen. So that prosthetic group is attached through a covalent bond, as you can see. Now the iron atom present is in the ferrous state and can reversibly bind to oxygen, carbon monoxide, and nitric oxide. The alpha 1, beta 1, and alpha 2, beta 2 dimers are connected by a two crease pivot symmetry, and the interfaces of the subunits alpha 1, beta 1 and alpha 2, beta 2 are identical. And alpha 1 beta 2 and alpha 2 beta 1 subunit interfaces are mirror images. So that's the idea. So beta 1 and beta 2, these are mirror images. Alpha 1 and alpha 2, these are alpha 1 and beta 2, if you see diagonally, they are mirror images. So that's what they are writing. And alpha 1 beta 1 and alpha 2 beta 2, these are identical. So that's the symmetry that they have. Now if you talk about the human globin gene cluster, then the synthesis of hemoglobin is controlled by two developmentally regulated multi-gene clusters. One is the alpha globin gene cluster on chromosome 16, and then there is the beta globin gene cluster on chromosome 11. So the alpha globin gene cluster, it has three functional genes. So there is the zeta gene, and then there is the fetal or adult alpha 1 and alpha 2 genes, as you can see in the slide. Then the beta globin gene cluster, it has five functional genes. So that is the answer to the question. So it has the embryonic gene, the HBE. It has two fetal, gamma and genes, G gamma and A gamma. And then there are adult genes, which are delta and beta, which are present in the human beta globin cluster. So that's how it looks like. Now we come to the question number 3. According to the degree of quantitative reduction in the production of beta globin, beta thalassemia alleles are classified into three categories. Which of the following is not in the category? Is it A, absence of beta globin, or B, 0? Beta globin is produced but reduced, or is it beta globin production is minimally reduced, or is it delta globin is produced and increased? So I'll give you a minute to think on this, and then we'll discuss the answer. Okay, so let's discuss the answer. According to the degree of quantitative reduction in the production of beta globin, beta thalassemia alleles are classified into three categories, out of which the one which is not an option is D, delta globin is produced and increased. So let's see. So what happens in beta thalassemia? There are two in a normal cell. There are two copies of beta globin gene. They are present in chromosome 11. Now, if one copy of beta globin gene becomes impaired, then mild anemia might arise. If both the copies are impaired, then there is severe anemia and a potentially life-threatening condition. Now, beta thalassemias are majorly caused by non-deletion type mutations, including single nucleotide substitution, short insertion deletions leading to frame shift mutations. So we have already discussed about frame shift mutation earlier when we were discussing about damages to DNA. So deletion in beta globin gene upstream locus, in the upstream locus region, LCR are rarely seen in beta thalassemias. So let's see. So according to the degree of quantitative reduction in the production of beta globin, beta thalassemia alleles are classified into three categories. It could be due to the absence of beta globin completely. So the beta globin is not produced at all. That would cause beta thalassemia. Beta globin is produced, but it is, not produced as much as is required. So that could be there. Reduction in the production of beta globin, or beta globin production is reduced, but it's reduced minimally. that could also happen. These could lead to beta thalassemia. So based on the clinical presentation, alpha globin or beta globin chain imbalance, severity of anemia, and RBC transfusion requirement, beta thalassemias are classified into beta thalassemia minor or beta thalassemia trait patient. Okay, so if there are individuals with heterozygous mutation, there is a wild type beta globin and a mutated beta globin. In that case, we have such beta thalassemia minor. If there are clinically asymptomatic mild to moderate hypochromic microcytic anemia, if it is there, that could happen. Now they do not require RBC transfusions, so they are slightly anemic, but they can live normally. Mild erythroid hyperplasia in bone marrow with normoblasts showing poor hemoglobinization. Mild to moderate splenomegaly. Splenomegaly is enlargement of the spleen that happens in very small individuals because there is a regular expressing beta globin gene that compensates for any lack of gene. So some patients may not show and any hematological abnormalities and are termed as silent carriers, but these silent carriers can definitely uh, cause this beta thalassemia to happen to their offsprings. So they can definitely make that happen. So beta thalassemia could be of beta thalassemia major also, where there is mutation in both the beta globin genes, and there is severe anemia. There is these are homozygotes or compound heterozygotes for beta globin mutation may occur in intermediate or major. Intermediate tend to present later in life with mild to moderate anemia and symptoms. Intermedia is a rare clinical phenotype where a less severe alpha to non-alpha globin subunit imbalance occurs, hence less accumulation of free alpha subunits. In major, a severe alpha to non-alpha globin subunit balance is present, resulting in high levels of accumulation of free alpha subunits, but there are not multiple beta subunits. Now, more than 350 disease-causing mutations have been identified so far. You can see the epidemiology of beta thalassemia. It's given in your lectures. So majority of beta thalassemia phenotypes are due to mutation in one or more nucleotides in the beta globin cluster, or instant flanks that result in shift in the reading shift. Open reading frame is shifted. Cross-gene deletion in beta thalassemia were reported in rare conditions. Beta thalassemia mutation may occur in exonic, intronic, or the promoter sequences, and they affect the beta globin gene expression. Any type of missense mutation, minor deletions of frameshift can result in hyper unstable beta chains, which are then further degraded through the normal, proteostasis machinery. Now, mutations are producing in typical recessively inherited forms of beta thalassemia located in exons 1 and 2, mostly in exons 1 and 2, and in exon 3 mutation in the phase termination codons result in dominant beta thalassemia, in contrary to the classical recessively inheritance beta thalassemia. So if there is a mutation in exon 3, there could be a dominant form of beta thalassemia, where, although it is very, very, very rare. But usually beta thalassemia is also a recessive disorder. So let's talk about the next question. DASH is the major factor in switching of gamma to beta globin production in adulthood, which activates expression of BCL11A, SOC6, and ZBTB7A to inhibit expression of gamma globin gene. Is it KLF1, BCL1, GATA1, or HPFH1? So I'll be giving you a minute to think about it, then we'll discuss the answer. Okay, so the answer to this question would be KLF1. It's a major factor in switching of gamma to beta globin production in early adulthood. So let's see how developmentally this is regulated. So if we talk about the gene editing, just give me a moment. Yeah. So we see the gene editing in thalassemia. So there are, therapeutic genome editing methods to use the homology directed repair pathway for genetic repair of the or the non-homologous end joining pathway. There could be two pathways, as we have already discussed. For genetic disruption using the programmable nucleases like zinc finger nucleases, TALENs, and CRISPR-Cas9, we have already discussed how these work. The strategies to tackle thalassemia using gene editing is one of them is HbF is targeted, or gamma globin induction using gene editing tools. So HbF is secreted during the fetal stages. So if we can somehow induce this gene to secrete, then the absence of beta globin can be compensated. So HbF repressors could be targeted. Then we could introduce HPFH mutations that recapitulates a mutation associated with a benign genetic condition. Or we can have some gene repair strategies where the mutation in the beta globin gene can be repaired. So let's look at it. So down regulation of gamma globin gene is normally controlled by several epigenetic modifications and transcription factors, and it is also developmentally regulated. So that's the thing. By targeting the fetal hemoglobin repressors, BAF chromatin remodeling complex subunit, BCL11A, SRY box transcription factor 6, SOX6, HBS1L, MYB, KLF1, and zinc finger and BTB domain containing 7A. So that is ZBTB7A. The synthesis of fetal hemoglobin can be restored. So using these repressors, the fetal hemoglobin production is lowered. But if we actually target these repressors and make them non-functional, then the idea is the fetal hemoglobin levels will be restored in the blood, and that would compensate for the lack of beta globin in the blood. So the genome editing techniques like zinc finger nucleases, TALENs, it would be TALEN, sorry for this error, it would be TALEN, and CRISPR-Cas can be effectively used to reduce or delete expression of the HbF receptors. However, various types of side effects may also come up, since transcription factors often have a broad spectrum of functions to regulate multiple target genes. So it's not like a particular a transcription factor only regulates a particular gene. They might regulate multiple genes. So if you down-regulate a transcription factor, it might lead to some deleterious effects. So let's see what happens. What we do in adults, binding of BCL11A and SOF6 and ZBTB7A to the proximal promoter of gamma globin cause it to repress. So when it binds to gamma globin, it causes repression of gamma globin. So KLF1 is a major factor in switching of gamma globin to beta globin production in adulthood, which activates expression of BCL11A. So this is the cyclic process which inhibits the expression of gamma globin gene. KLF1 as an activator also upregulates beta globin through direct binding to it as a promoter. Now, HbF upregulation can be achieved by targeting erythroid-specific elements using zinc finger nucleases, TALENs, or CRISPR in KLF1, SOX6, BCL11A, and ZBTB7A gene, and their expression is attenuated. Downregulation of KLF1 and BCL11A transcripts lead to increased gamma globin mRNA expression and considerable fetal hemoglobin production. However, targeting KLF1 is not preferable as it has implications in other biological pathways as well. So that's how the answer is. So disruption of the GATA1 motif in the erythroid-specific intronic enhancer of BCL11A achieved through nucleases or cytosine-based editors determines BCL11A knockdown and consequently reactivation of gamma globin. So that's how it is done. So in HPFH mutation called British HPFH, sorry, HPFH mutation called British HPFH is caused by a point mutation of the dash in the promoter of the fetal globin gene. So is it 198C to T, or is it 198T to C, 198G to C, or 198T to A to T? So I'll give you a minute to think about it, and then we'll discuss the answer. Okay, so the answer to this question would be 198T2C. So let's see. So reproducing the HPFH mutations recapitulates a mutation associated with a benign genetic condition. Let's see how it happens. So HPFH stands for hereditary persistence of fetal hemoglobin. HPFH stands for that. It's a disorder that happens when the fetal globin promoter sequence, it develops some mutations that cause fetal hemoglobin to reactivate in adult life. So that happens, that's a disorder that happens to few people, but we need to use this disorder to the advantage of beta thalassemia patients. So there are small deletions or single point mutation in the proximal promoter of the fetal gamma globin gene that cause these natural mutations. Recent attempts have been made to simulate these mutations to reactivate the fetal hemoglobin in thalassemia patients as a therapeutic strategy. So by creating a point mutation mimicking the hereditary persistence of fetal hemoglobin phenotype via targeting the binding sites of the repressors BCL11A and ZBTB7A to inhibit their binding to gamma globin gene promoter, the HbF expression can be upregulated. So let's see how that works. So in case of the British, HPFH, there is a point mutation at the 198 position, which causes a T to C mutation in the promoter of the fetal globin gene. Now, the symptoms of hemoglobinopathies are significantly reduced when they're in the thalassemic patients who have this kind of mutation. And the fetal hemoglobin production, it increases by 20%. In an in vitro and in vivo combination study by Wienert and his colleagues, CRISPR-Cas9 mediated homology-directed repair system was used to simulate this mutation, 198T2C, uh, in the fetal hemoglobin promoter of the clonal wild-type HUDEP2 cells for mimicking the natural mutation. This led to the creation of a new binding site for KLF1, the major erythroid gene activator, which further enhanced the fetal hemoglobin levels by elevated the mRNA expression level of gamma globin. So this study has recently been done in 2022. So that's how it is. So another type of mutation also naturally occurs that also can be mimicked. So that is a 175T2C single point mutation, which leads to a significant increase in the fetal gamma globin to erythroid cell lines ratio. So the elevated fetal globin level in these individuals is related to de novo recruitment of the activator TAL1 to promote chromatin looping of distal enhancers to the modified gamma globin. In a study, the 175T2C substitution into A gamma and G gamma globin generates promoters of murine erythroid and human erythroid K562 cells was induced using talin and homologous recombination strategies. In both the cell lines, TAL1 could bind to the promoters and they could activate the gamma globin in the mutated erythroid cells. So the percentage of fetal hemoglobin, it increased from 16 to 41%. of total hemoglobin. So here the increase is much higher than in case of the previous mutation. Now, other point mutations could also happen in the BCL11A and the ZBTB7A genes that could also cause reinduction of HbF. So in wild type, these two transcription factors, they bind with the gamma globin gene promoter at the gene positions 115 and 200 base pair respectively, and they repress the fetal globin gene expression. Martin and his colleagues used CRISPR-Cas9 technology to induce a homozygous HPFH-associated mutation of 117G2A, 114C2A, and a deletion of 13 base pair, and 195C2G in the gamma globin gene promoter of the erythroid cells, where BCL11A and ZBTB7A usually binds. So it is because of this mutation, these transcription factors, which are actually repressors, they could not bind to this gamma globin gene, and they cannot repress the gamma globin, and hence the fetal hemoglobin levels are increased. So that's what it is there. So let's come to the next question. Natural killer cells are the predominant innate lymphocyte subsets that mediate A, anti-immune and antibacterial responses, B, anti-tumor and antifungal responses, C, antihistamine and antiviral responses, or it is it D, anti-tumor and antiviral response. I'll give you a minute to think and then we'll discuss the answer. Okay, so the answer to this question would be natural killer cells are the predominant innate lymphocyte subsets that mediate anti-tumor and antiviral responses, option number D. So let's see. The T cells they derive from, let's study a little bit about the immune cells. So the T cells they derive from the hematopoietic stem cells in the bone marrow, and they follow migration and they mature in the thymus. That's why they are called T cells. These cells express a series of unique antigen binding receptors on their membrane known as the T cell receptors. Each T cell expresses a single type of T cell receptor and has a capacity to proliferate and differentiate if it receives the appropriate signals rapidly. As previously mentioned, T cell required action of antigen presenting cells. They are usually dendritic cells and macrophages and B cells, fibroblasts, epithelial cells to recognize a specific antigen. So the idea is the antigen presenting cell, they will digest the pathogen, they will present the pathogen, and they will in a type of antigen, and then these antigen presenting cells would be recognized by the T cells. That's the idea. Now, B cells, they arise from the hematopoietic stem cells in the bone marrow, and they follow maturation, leave the marrow expressing a unique antigen binding receptors on their membrane. Unlike T cells, B cells can recognize antigens directly. They do not require any antigen processing through unique antibodies expressed on their cell surface. The principal function of B cells is the production of antibodies against foreign antigens, which require their further differentiation. Under certain circumstances, B cell can also act as antigen presenting cells. So about natural killer cells, they are predominant innate lymphocyte subsets that mediate anti-tumor and antiviral responses. So that gives us the answer. Natural killer cells recognize and kill cancer or virus-infected cells and therefore possess promising clinical utilization. And one of the best things about these kind of cells is that these are present as a part of the innate immune system. Now, natural killer cells accounts for 5 to 20% of the mononuclear cells and the peripheral blood and spleen. They produce cytokines along with interferons, and among which interferon gamma delivers signal to the innate component of the immune system, which activate the inflammatory process in the defense of the organism. So if there is a virus or if there is a tumor, virus infected cell or a tumor infested cell, that would be, killed using this inflammation. So the activation of natural killer cell function following interaction with a target cell is the result of the integration of signals generated by inhibitory and activating receptors expressed simultaneously by natural killer cells and engaged by the ligands present on the target cell. Natural killer cells are fundamental as a defense mechanism as they can act early during a cell infection or transformation before and independently of specific immunity. They take part as the first line of immune response or the first line of defense. That's why they are innate immune cells. Now, coming to question number 7, in the first trials of gene therapy for SCID, severe combined immunodeficiency, retroviral vectors were used in which expression of the normal transgene uh, was driven by retrovirus long terminal repeats, LTR, or self-inactivating retroviruses, SYN-RV, graft versus host disease, or immunoglobulin replacement therapy. I'll give you a minute to think on this and then we'll discuss the answer. Okay, so the answer to this question would be in the first trials of gene therapy for SCATE, retroviral vectors were used in which expression of the normal transgene was driven by a retrovirus long terminal repeats. So let's see. In case of gene therapy with patients, hematopoietic stem cells was developed to tackle the problem of graft versus host disease and a graft failure rejection associated with transplants. So the first gene therapy trial on 14 September 1990 was carried out on Ashanti De Silva for SCID. That's why it's called ADA kit. Now, in gene therapy, autologous hematopoietic stem cells, they were harvested from the patients and are transduced with a viral vector that contain a correct copy of the gene along with the regulatory elements that control gene expression, such as promoter and enhancer. So the viral vector allows the integration of these therapeutics transgene into the hematopoietic stem cell genome, and corrected hematopoietic stem cells are infused back to the patient, right? So that's the idea. If you see, in this case, the CD34 positive cells, so they are sorted from the bone marrow or mobilized peripheral blood stem cells are used using magnetic separation with culture and cultured with cytokines. These cells are transduced with the specific vector containing the corrective human interleukin 2 receptor G IL2RG gene, and after a period of culture, these cells are reinfused into the patient. In the most recent clinical trials, the cells are infused after a low dose conditioning treatment. Subsequently, the immune reconstitution starts and the patient is theoretically cured for life, although we do not have such a gene therapy based approach yet that is used for medicine. In the first trials of gene therapy for SCID, retroviral vectors were used in which expression of the normal transgene was driven by retrovirus long terminal repeats. So that gives us the answer. With this approach, successful and durable T cell reconstitution was achieved in patients with XKID and ADA-SKID. Unfortunately, several patients develop leukemia due to preferential integration of retroviral vectors in proximity of the transcription initiation sites of the genes, including the oncogenes, and by the strong enhancer activity of viral long terminal repeats, leading to increased and deregulated expression of the targeted oncogenes. So what happens? Retroviral vectors, lentiviral vectors, these are integrating viral vectors. So what happens when they get into the cell? They can integrate into the virus genome. And this integration process is not controlled. It could happen in a very important gene. It could happen in a oncogene. So in such a case, the regulation of the cell will be severely disrupted because they have these long terminal repeats which have the ability to enhance the expression. So let's say if they are not they are not getting integrated in the right place, then that creates a problem. To overcome the above problem, we have self-inactivating retroviral vectors in which U3 enhancers was deleted from the LTR and expression was driven by a weaker eukaryotic elongation factor, EF1 alpha. Short promoter was developed as a safe alternative. Additionally, SYN lentiviral vectors have been developed to reduce integration within proto-oncogenes. So in the first generation gamma retroviral vectors, the gene expression is controlled by a viral long terminal repeat. So that you can see here. That is how they control the gene expression in the A part. In the second generation, self-inactivating gamma retroviral vectors and lentiviral vectors are used where the U3 region of the LTR is deleted and the human interleukin um, 2 receptor gamma gene, IL2R gene expression is derived by an internal mammalian promoter. Moreover, lentivirals contain a codon optimized IL2RG complementary DNA to further improve the transgene expression, and U3 region is replaced with a chromatin insulator element. So that's the answer to the previous question. Let's go to question number 8. Hemophilia is usually an inherited bleeding disorder in which the blood does not A, clot properly, B, bleed properly, C, generate properly, or D, repair properly. So I'll give you a minute to think on this and then we'll discuss the answer. Okay, so the answer to this question would be hemophilia is usually an inherited bleeding disorder in which the blood does not clot properly. Let's see how what is hemophilia. So it's usually an inherited bleeding disorder in which the blood does not clot properly. So that gives us the answer. This can lead to spontaneous bleeding as well as bleeding following injuries or surgery. Blood contains many proteins, which includes the clotting factors which prevent bleeding. Now, people with hemophilia have severely low levels of either factor 8 or factor 9. The severity of hemophilia that a person has is determined by the amount of factor in the blood. The lower the amount of the factor, the more likely it is that bleeding will occur, which can lead to serious health problems. In rare cases, a person may develop hemophilia in later in life. In the majority of cases involve middle-aged or elderly people or young women who have recently given birth or are in the later stages of pregnancy. This condition often resolves with appropriate treatment. So let's see a little bit more. So it's a hereditary bleeding disorder, and anyway, these hereditary bleeding disorders are a group of multiple diseases that include inherited abnormalities of primary and secondary hemostasis. It results from the deficiency or functional abnormality of one of the plasma proteins that are involved in the coagulation of the blood. So essentially, clotting factors. So hemophilia and von Willebrand syndrome, VWD, are the most common types of these hereditary bleeding disorders. About 4 lakh people are suffering from hemophilia, and only 25% of them receive adequate treatment. This is according to a paper in 2019. Hemophilia is characterized by painful and often spontaneous hemorrhages into joints and soft tissues that are life-threatening if it is intracranial, gastrointestinal, or in the neck or throat region. So hemarthrosis accounts for 70 to 80% of all bleeding episodes and lead to hemophilic arthropathy. So hemophilic arthropathy finally leads to major issues with joint mobility, decreasing overall patient function and the quality of life. Now let's see how it is inherited. So it is caused by a mutation in one of the genes that make the clotting factor proteins that are needed for the blood to clot. And this is an X-linked recessive disorder. So it involves the X chromosome. So such genes are located in the X chromosome. The males inherit the X chromosome from their mothers and the Y chromosome from their fathers. The female inherit one X chromosome from each parent. So naturally, because of this, since this disease is an X-linked disease, so a male is more likely to be affected. Apart from that, a daughter, if affected, usually do not live for long because they have more blood loss compared to males. So that's the thing. So hemophilia is classified into three: hemophilia A, B, and C based on the factor which is deficient. So hemophilia A has factor 8 deficiency, hemophilia B has factor 9 deficiency, hemophilia C has factor 11 deficiency. So hemophilia A is also called a classic hemophilia and is the second most common type of all hereditary bleeding disorders after the VWD and the most common type of hemophilia in the world. Hemophilia A is 5 times more common than hemophilia B. Hemophilia A and B are the only HBDs that are linked in a sex-linked model, and our affected gene is located on the long arm of X chromosome. Hemophilia C is the rarest form. It's an autosomal repeating disorder, and bleeding symptoms due to the deficiency of factor 11 are seen in hemophilia C. So the frequency of bleeding, it depends on the residual coagulation factor level and the genotype of the patient. Factor 8 and factor 9 level, which is normally present in around 50 to 150 units per DL deciliter, typically correlates with bleeding severity. So there could be mild hemophilia where the levels are 5 to 50. So bleeding is more likely to occur following a surgery or trauma, and unprovoked hemorrhages are rare. In moderate hemophilia, the factor levels are from 1 to 5 units per deciliter. So the hair bleeding is usually observed after injuries, but spontaneous bleeding episodes without any reason also occur. In severe hemophilia, where there is absolutely almost no factors, the patients, they experience recurrent spontaneous bleeding effect with hemarthrosis, bleeding in the muscles, soft tissues, and other life-threatening bleeds, like intracranial hemorrhage, as well as excessive bleeding during and following surgery or trauma. So recurrent hemarthrosis leads to hypertrophic synovitis, progressive cartilage degradation, and hemophilic arthropathy characterized by chronic pain, severe deformity, and reduced mobility. Patient with non-severe hemophilia can also suffer considerable morbidity and increased mortality risk, because if they get into some accident, they will die sooner because their blood doesn't clot normally. Even in patients with mild hemophilia, the mean number of bleeding episodes is 0.44 to 4.5 per year, which severely interferes with the quality of life. Males with hemophilia have a lower life expectancy than the general male population, even after treatment-related improvements. The decrease in life expectancy of patients with hemophilia A in developed countries is 30%, while those with severe hemophilia A is 37%. So let's talk about another question. The F8 gene, that is the factor 8, is a large gene that is comprised of A, 10 exons, B, 25 exons, C, 30 exons, or D, 26 exons. So I'll give you a minute to think on this and then we'll discuss the answer. Okay, so the answer to this question will be F8 gene is a large gene that comprises 26 exons. So let's look at factor 8. So as we have already discussed that factor 8 deficiency causes hemophilia A. So they suffer spontaneous, and it's one of the most common forms of hemophilia. So they suffer spontaneous bleeding events, including those that are life-threatening and develop joint damage as a result of recurrent bleeding into the joints. Hemophilia A is caused by decreased activity of plasma coagulation factor 8 due to mutations in the F8 gene encoding this protein. The human F8 gene is localized on the long arm of X chromosome and consists of 26 introns and exons for a total length of 9 kilobase pairs in the coding sequence. Sinusoidal, endothelial, and Kupffer cells in the liver are the major site of F8 expression. Now, the gene encodes a large precursor glycoprotein of 2,332 amino acid residues and consists of 6 structural domains and 3 acidic subdomains organized in a heavy and light chain. Every domain plays a physiological role throughout the life cycle of factor 8, from biosynthesis to clearance. The B domain, a 908 amino acid residue long region, is not required for F8 clotting activity, but likely they do have a role in processing intracellular transport and secretion of the F8 protein. So this is how the F8 factor looks like. Now, the B domain is partially removed from the mature factor 8 so that several truncated B domain variants of F8 circulate in the bloodstream. Complete or partial deletion of B domain are not associated with significant decrease in procoagulant properties of these F8 variants. Post-translational modification of F8 precursor enables sulfation of tyrosine residues by sulfotransferase in the Golgi apparatus. So there are 6 potential tyrosine sulfation sites on the F8 molecule, that is 4 on the heavy chain and 2 on the light chain. All the 6 sulfation sites are required to modulate this activity of F8, and the sulfation of key tyrosine residue is crucial not only for the function of the clotting factor, but also for its stability and binding to the von Willebrand factor. So F8 plays a very major role. So here you can see the structure of F8. So the domains are in capital letters. The subdomains are in lower cases. Glycosylation sites are denoted by triangles. Disulfide bonds are denoted by arcs. And reduced cysteine residues are denoted by orange vertical lines. The S inside the circle indicates sulfated tyrosine. Residues bracket mark the area of interaction, uh, and so on and so forth. You can study about the structure. So let's see the structure of the gene. So the F8 gene is a 186 kilobase pairs gene which consists of 26 exons. So that gives us the answer. The 24 of the exons, they vary in length from 69 to 262 base pairs. Exon 14 is of 3106 base pair, are much larger than the other exons. Most of the 26 exon consist of a 3' untranslated sequence. Intron 22 contains two further genes, F8A and B, with transcription orientation in the opposite directions, controlled by a bidirectional promoter. So this comes, we're coming to the last question of the day. The ASC618 construct uh, consists of adeno-associated viral vector encoding a codon optimized HF8 gene that has been minimized by deleting the AH domain, the B domain, the CA domain, or the DC domain. So I'll give you a minute to answer this. We have already discussed, we have already given a hint to the answer. So let's see. You can think about it, then we'll discuss. Okay, so the answer to this question would be B domain. So we have already discussed that B domain is involved. For not, it does not affect the stability or the activity of the factor 8. So that's why we can delete it. So let's see. So for gene therapy, since the large size of native factor 8, it is difficult to package in a clinically applicable vector. However, deletions of sequence encoding hemostatically non-functional domain B domain deletion of the F8 cDNA has allowed its incorporation in adeno-associated vectors, viral vectors. The first successful application of this approach was reported in 2017 using an AAV5 co-BDD-F8 vector. So we'll see how this vector looks like in a dose escalation study in 9 patients. Applications with high dose cohort, the F8 levels was found to increase greater than 5 units per DL by weeks 2 and 9, rising to the normal range in 6 to 7 out of 7 patients after week 20. The stability of F8 expression was achieved at 1 year median, which reduced annualized bleed rate from 16 events per year to 1 event per year for the patients. So increase in the serum levels of alanine transaminase that indicate liver toxicity occurred in 8 out of 9 participants without an associated cellular immune response, managed with corticosteroids without change in F8 transgene expression. So let's see how this cassette looks like, this expression cassette. So this is the ASC618 construct that consists of an adeno-associated viral vector, AAV8 vector, encoding a codon optimized F8 human F8 factor that has been minimized by deletion of the B domain. The construct is designed to be specifically expressed in liver cells. So let's see what are the disadvantages of using an adeno-associated viral vector. So the first one is viral immunogenicity. So there could be, neutralizing antibodies against the virus. It is recognized by the immune system of the host, and when there is subsequent introduction of the same viral vector, then there is immunity that causes degradation of the virus and it doesn't function. So potency and efficacy is an issue, and that's an issue with major gene therapy applications. So the efficiency with which adeno-associated vectors can infect and transduce into the desired targets, they can impact the therapeutic doses and efficacies. There could be genotoxicity, although it's very rare. The integration of AAV vector DNA into the genome of the infected cell may cause some genotoxic effects. Sometimes cells also die due to the viral load. And the persistence, the episomal adeno-associated vector genome in the nucleus of the infected cell can be lost in conditions of cell proliferation. So again, if if the cells are proliferating and they are not transmitting the corrected virus, then in that case we won't have a continuous expression of clotting factors. So that would cause into a loss of clotting factors with time. So that's the idea. That is all that we have today for today's question answer sessions. If you have any doubt, you may ask, or we may just go through the questions once again if anybody has missed. Uh, okay, we'll be meeting again next week on the same time. Just before, if anybody has left anything, we will just discuss the answers of all the questions once again, and then we will call it a day. So coming to question number 1, that is, thalassemia is a hereditary disorder and it is inherited by an autosomal recessive manner. Then there is question number 2 that says the functional genes that are there in beta globin gene clusters. There are 5 functional genes. Then coming to question number 3, according to the degree of quantitative reduction in the production of beta globin, beta thalassemia alleles are classified into 3 categories. Which of the following option in this is not the category? So the option D, that delta globin is produced and increased, is not an option in the category. So then we have question number 4. So KLF1 is the major factor which is involved in switching of gamma to beta globin production in adulthood, which activates BCL11A, SOC6, and ZBTB7A to inhibit the expression of gamma globin gene. Then we have question number 5. HPFH mutation called British HPFH is caused by a point mutation of the 198T2C in the promoter of the fetal globin gene. So that's there. And then we have the question number 6, that the natural killer cells are predominant innate lymphocyte subsets that mediate anti-tumor and antiviral responses. Then we have question number 7. In the first trials of gene therapy for severe combined immunodeficiency, retroviral vectors were used in which the expression of the normal transgene was driven by the retrovirus long terminal repeats. So after this, we have question number 8. Hemophilia is usually an inherited bleeding disorder in which the blood does not clot properly. Then we have question number 9. The F8 gene is a large gene that comprises 26 exons. And then the final question is that the ASC618 construct consists of an AAV8 adeno-associated viral vector encoding a codon optimized human factor VIII gene that has been minimized by deleting the B domain. So that's all for today's question answers, live interactive session. If you have any questions, you may ask, or we may be meeting again next Tuesday.
Thank you, sir.
Okay, thank you. You may leave if it is done.